In this video, we're going to finish up looking at the ways that microbiologists determine evolutionary relationships between the bacteria that they're studying. And if you recall, the last video we were talking about genetic homology, or similarity in the genetics of a bacteria that are being studied, and using the similarity as a proxy for how closely related the bacteria are. So as we're thinking about genetic homologies, another data point that we can gather is looking at how much there is of each of the four bases in the DNA of the bacterium. So DNA is built of what are called nucleotide bases, and there are four of them. There's adenine and thymine and cytosine and guanine. So these four bases are what makes up the genetic code. And the A's and the T's always connect together when we're building the double helix. So if we had A, G, T, C, A, T, if that was our sequence on one half of the double helix, then on the other half we'd see T and C and then A and T and G, sorry about that, and T and A. So if two bacteria have exactly the same DNA, then they're going to see, if you could separate out what percentage of these nucleotides are one or the other, then you could you would have a way of looking at similarities. And so if two bacteria had exactly the same DNA, then their CG content, so the amount of cytosine and guanine that we find in there, and the ratio here would be, it's like 1, 2, 3, 4, 5, 6. So what is that, about 18% each? So this would be 36%, give or take, CG, and then the other 64% would be AT. And so if you could find a bacterium that also had 30% GC, then while that's not complete proof that you have exactly the same sequence of DNA, because they could have a different order of these DNA, but it gets you a little bit closer. It tells you, yes, there's enough similarity here between these two that we should investigate them further as either being identical or being closely related. Or, on the other hand, you could say, whoa, the ratios are way out. Now, if the ratios are way out, then there's no way, if they're very different from each other, then you would know emphatically that they were not closely related to each other. So this percentage of CG that we find, or think of it as the CG ratio or percentage, that is going to be a test that you can use to discriminate between closely related and, or at least to say that the bacteria are not very closely related. So across bacteria, we see CG content rates from as low as 23% of the DNA to as high as 75%. So if you had one bacterium with, say, a 45% CG content, and another bacteria with let's say a 28% CG content, then you would have a lot of reason to say that these are not closely related at all. Whereas if you saw 45 and 45, then that would be evidence that perhaps they could be very closely related. Okay, so that's the relative abundance of the nucleotide bases is one way as a proxy for genetic homology. A second is looking at DNA and RNA sequences of very short strands. So we can send in enzymes that will clip up the DNA and the RNA, and then use and then melt them, separate them from each other, and then send in these little strands of DNA or RNA that we build that have that would be the pair up for the DNA that we're looking at. And if you tie a little fluorescent dye or a bit of radioactivity to these probes, then you can send the probe in, introduce it to the sample of DNA bacteria, let the two give it time for these little chunks to mate up, and then rinse everything off, so you wash away any unmated bits, and then look for fluorescence. Put this under a microscope and look for fluorescence, or look for radioactivity. And that can then be used, if there's similar amounts of fluorescence or radioactivity in the two samples of the different bacteria you're looking at, then you would have reason to believe that we have very similar DNA sequences. If there is very little similarity in the amount of fluorescence or in the amount of radioactivity, then we'd start thinking, okay, these are definitely different from each other. Okay, so we can use short bits of DNA and RNA to go in and pair up with the bacteria's DNA and RNA and tell us how much there is of those particular sequences. So you can do another process called DNA hybridization. So here you would take DNA from the two different bacterial species that you're interested in, and then through a process warm it up. This is called melting the DNA. With a little heat, you split the double strand of DNA so that they are no longer attached to each other. And if they're all mixed in together, then when you cool it back down again, the strands will bond back together with each other. And they may bond with a partner from their own bacterium, or they may bond with a partner from the nearby, or they also blended in there, second bacterium that you're checking to see if there's a lot of genetic similarity to. If the true strands anneal completely, and annealing means letting them bond back together again, if you get 100% annealment, then you know, woohoo, we've got the same species. But if you see just a little bit of regions where the DNA don't anneal, then perhaps that tells you that maybe these are related, but not very, but they are. But there are some differences going on. Well, there doesn't tell it directly measures for you whether or not there are differences. And if there's very little annealing that occurs, then that tells you that they are definitely not related to each other. So the less annealing or the less rebonding together of these two different species of bacteria or two different groups of bacteria that you're comparing, the less that they bond back together, then the more distant they are related to each other. The more we see annealing, then the more likely it is that they are closely related. So that's DNA hybridization, because you're forming hybrids between the two different species or two different bacteria that you're trying to get information about. So if DNA is the instructions for making proteins, and then you could look at the DNA directly and look for similarities, or you could look at the proteins and look for similarities in the proteins. So for every 3 nucleotide bases that are in a string of DNA, that is a code for 1 amino acid. And of course, proteins are very, very long, but if we have proteins and we have proteins from 2 different bacteria, we keep them separate from each other, we're not blending them together this time, and then use enzymes to chop the proteins up into shorter strands between very specific sets of the amino acids, then what we would expect is that if the bacteria proteins are coming from the same piece of DNA, then those bacteria proteins are going to be chopped up into the exact same little segments. If, on the other hand, the bacteria proteins are coming from different pieces of DNA, then, so very different DNA, and therefore coming from likely different species or different, more distantly related groups of bacteria, then you would expect their proteins to be chopped up into very different chunks. And then, so you would take and isolate the proteins from the bacteria that you're interested in, and then chop them up using enzymes. And then load them into an apparatus called an electrophoresis gel. So electrophoresis gels are built so that you can pour this gel solution. It's called acrylamide gel. So you use this material called polyacrylamide. So once it cools, it sets up like gelatin does, and you build into it. So as you're forming this gelatin, you build in these little wells, and you can see them as like the empty spaces between the chunks, like at the top of a castle wall. So into those empty spaces, you can put your digested protein, the protein that's been subjected to the enzyme, that's going to have all these different length strands of protein in it. And remember, if they are similar to each other, then you're going to see similar lengths of proteins, and if the bacteria are very different genetically, you would expect to see chunks of proteins that are of different sets of lengths. So then you use an electric current to drag those proteins through the gel. And the smaller the protein is, then the easier it can dodge amongst all the gel molecules, and it travels really quickly. Large proteins, on the other hand, they take a lot more time to pass through this maze of gel molecules, and so they don't travel as far. And so you do this process of electrophoresis, where you separate all the different lengths of protein, amino acid segments of the protein. You drag them through that gel, and they separate out by their length. And then, after you're all done with that, you turn off the electricity, pull out your polyacrylamide gel, and then you stain it. So here is an example of one that's been stained with a blue dye. Come on, there we are. And you see this pattern of bands. The bands here, so if this is where, if we loaded them in at the top, then the bands here at the bottom are showing you how much of the protein was cut into little tiny segments. And up here at the top, this is where the proteins were cut into really big, long chunks. And the whole spread here that we see along the length of that lane, that's how we describe that lane, is all the different chunks and the different lengths that they are. And you can run, in these other lanes here, you can run like a set of standards of known lengths of amino acid chains. And that will then give you an idea of how many amino acids there are in each of those chains. And then you can compare the run from a number of different bacteria. So here we have 3 samples from one bacterium, 3 samples from another bacterium. And you can see that bacterium A, all 3 samples show a whole lot of protein right here, another big band here, and then a band right there. Well, bacteria B has a very similar band of really long proteins, but then we see a really different set banding pattern here. Just very few, relatively speaking, proteins at this length, and then maybe a little bit more right down here at the bottom at this length. Okay, so there you have it. Using gel electrophoresis to get an idea of what the proteins in the bacterium are like as a proxy for looking at how similar the actual DNA is. Okay, and then there are other techniques that aren't related to genetic homology. So here's the last set of techniques that don't relate directly to homologies among the genetics. One, you can compare the ribosomes to each other. So there are 2 subunits to build a ribosome. One of them is called the 16S subunit, and it clips on with the other subunit when it's time to make a protein using a piece of RNA. So the 16S subunit is highly conserved. That means that if you do many changes to it genetically, or to the genes that build that, if you change them, then you wreck that 16S subunit, and if it loses its function, then that's going to kill the cell. So it can't stand to be changed very much. And so since it's highly conserved, if you do see changes, or if you see differences in the proteins and RNA that make up the 16S subunit, then that tells you that those two bacteria are probably very, relatively distantly related to each other. And so we can analyze the construction of the 16S subunit and use that as a proxy of how, or as a measurement of how closely related they are. If they're very similar, then we would expect that they're going to be more closely related than if there are some of these subtle differences that can be tolerated by the cell and that can be allowed in the 16S subunit. So if you see any differences at all, then you would suspect very distant relationships between the bacteria that have those 16S subunits in them. So that's using ribosomes. You can also use antibodies. So bacterial cells have on them identifier proteins. So as a part of the membrane, there are proteins in there that are saying, hey, I am this kind of bacteria, and no other bacteria has those particular proteins. Or only closely related bacteria are going to have similar sets of identifier proteins on their cell membranes. And so in the lab, we can build antibodies that will bind to these identifier proteins and then tag the antibodies with fluorescence or radioactivity. And so you would take the bacterial cells, you would subject them to the antibodies, so give them time for the antibodies to bind, and once the antibodies bind, they don't release, then you'd rinse them off, again, getting rid of any unbound antibodies, and then you could compare different bacteria that have been subjected to this regimen to each other, looking to see if they have similar amounts of radioactivity or similar amounts of fluorescence. And the more similar the fluorescence or radioactivity rates are, then the more likely it is that the bacteria are sharing lots of common identifier proteins. And the more common the identifier proteins are, then the more closely related we would expect the bacteria to be. And then there's one third technique that's used for identifying whether or not bacteria are closely related, and that is phage typing. So bacteriophages, and they're sometimes just called phages, so we frequently don't say bacteriophage, but phages are viruses that attack and insert their DNA or RNA into bacterial cells. And in the process, they generally kill those bacterial cells. And so if you have a broth solution of a bacteria, and then you spread that broth, so do a spread plate where you spread it across a petri dish, the agar on a petri dish, and then infect that plate with a bunch of bacteria. With a bunch of bacteriophages. Then you can determine a little bit about how the bacteria are treated by the bacteriophages. So you can use different kinds of bacteriophages, and you would lay them down in a very specific pattern. So you can see that here, everywhere on this picture that you see clearing in the lawn of bacteria that you're growing, and that could be one there. I can't tell because it's off to the edge and it's a little different color, but it likely is. So these are all different bacteriophages that were put down in with a little dropper. And so generally you have a tool that will do this for you, and you can load bacteriophages into little into little reservoirs and then kind of print the bacteriophages onto the petri dish. Now, you might say to yourself, what an odd little pattern to make. Well, actually, they also put in bacteriophages in all of these locations as well. And so there's this pattern of bacteriophages that's laid down, and then you can compare which of the bacteriophages caused clearing or produced what's known as a plaque on the bacterial lawn. So a plaque is an area where the bacteriophages killed the bacteria. So if this is bacteriophage 1, so here's 1, here's 2, here's 3, here's 4, this is 5, 6, 7, and so on, then if the bacteria all respond, so if you then make several plates of bacteria, and you see that in plate 1, all the bacteria responded to the exact same bacteria as they did in plate 2, then you would expect or suspect that plate 1 and 2 are probably closely related or even identical bacteria. If, on the other hand, the bacteria in plate 1 and plate 2 showed a really different pattern of plaque formation, then you would suspect, oh, okay, something different is going on here. These guys are not producing closely related, or they're not susceptible to the same bacteria, phages, and so you would expect that they are different bacteria. Hopefully that made sense. So, similar patterns between two different samples of bacteria, then similarities, genetic similarities is what you would expect. Very different patterns, then you would expect that they are not closely related to each other. Okay, so that's using phage typing. So then what do the microbiologists do with this? They take all of this information then that they gather from all these different sources of information, looking at numerical taxonomy, by investigating genetic homologies, and then examining the other features, the ribosomes and the responses to the phage typing tests. And they use all of this data to produce cladograms that attempt to show the relatedness, so that we can then see, that bacteria A is more closely related to bacteria C than bacteria B is related to bacteria C. That's what our cladogram here would tell us, is that the closer we are here, the fewer of these branch points that you have to count your way to, tells you how much more closely related they are to each other. And so when we see differences, and that's what these are, these branch points here they tell us that a new characteristic has evolved, then we assume that is divergent evolution, that at that point we had a common ancestor, and from that branch point new species were evolved from this common ancestor. And we call that divergent evolution, when two new species evolve from a common ancestor. And so some of the early divergences that we can build into the evolution of bacteria is the development of gram positivity and gram negativity. So the differences in the cell wall that surrounds the bacteria. That was an early divergence. And then within those two gram-positive, gram-negative groups that evolved early from a common ancestor, so if this is gram-positive and this is gram-negative, then there was further, I can't go back, but, yes, there I can go back. So then there is further divergence that occurred, and even more divergence beyond that. And the same thing is happening over here with the gram-positive bacteria. They are also diverging. And so that's how we go about constructing these evolutionary trees or cladograms to try to make sense of how bacteria evolve. Okay, so that's getting you a little closer to understanding some of the techniques that are used by looking at genetic homologies, by looking at numerical taxonomy, and looking at some of the metabolic processes and the metabolic end products like proteins that are built inside of the cells to tell us a little bit about their relatedness. So you know what to do. If you have any questions, reach out to me.